Hello, everybody, and welcome back. In this video, we are going to talk a little bit about chromosome number and structure, changes thereof. This accompanies Chapter 6 in the Snustad genetics textbook. As you can sort of see here from the table of contents in the chapter, this is sort of a technical chapter. It's got quite a lot of terminology as far as these general words, polyploidy and aneuploidy, but then specific examples of that. And it talks a little bit at the end about rearrangements of chromosome structure. As we go through this, the big kind of take home from this is a follow-on from the previous chapter. In the previous chapter, if you remember, we talked about sort of that pivoting in thinking from when Mendel and others had defined the concept of inheritance, the concept of um, parental passing on of traits, and this sort of early 1900s kind of movement into figuring out what part of the cell is doing that. And of course, we know now that that's the DNA that's located on chromosomes. And so we sort of introduced that in the previous chapter. This chapter talks about what happens if the chromosome number and structure changes and how we can detect that. So let's head off to the slides. And you can, of course, find the slides themselves in the description below this video. Okay, and here we go. So as we go through the slides, of course, here's our table of contents again. We start out with cytological analysis, and the point of this is just to sort of give you some context for where these pictures come from. This particular diagram starts with a blood sample, but the point is we get some cells that have nuclei. So in this case, we'd be looking at white blood cells primarily. We could do this with skin cells or a biopsy of some other sort of structural tissue in the body. In any case, we would culture those cells here, stimulate them to divide, and importantly, disable the mitotic spindle. The point of doing that is to synchronize all of the cells in the same part of mitosis, excuse me. So the chromosomes have condensed, and we can see instead of the sort of diffuse DNA staining that you'd get from an interphase cell, we now will get the sort of chromosomal structures that you're used to thinking about. We then burst the cells here. We do that really easily with hypertonic solution. So water rushes in and bursts them. In this very technical term, squash. We make a slide, fix that, and look at the chromosomes. This is a picture from the 1970s. You can see in the caption there, using one of the dyes that was really common. This is one of the original fluorescent dyes, quinacridone. And you can see sort of not much. You can see a little bit of banding. Maybe you could pick out some patterns back then. It was fairly difficult. We, of course, have better techniques now. There are other dyes that stain chromosomes either visually or fluorescent. And one of those techniques, I'll show you in just a minute, improves our resolution quite a bit. So hopefully you can see here that these GIMSA-stained chromosomes look better. This paper is also from the 70s using a different technique. What this particular picture would be showing to the clinical researcher is that this chromosome on the left is present in 2 copies, that although they're curved, you can sort of see the banding patterns are pretty similar. The chromosome shown in the middle, again, duplicated. There might be a little bit of difference here. It's hard to tell. But clearly, this 3rd chromosome has had a deletion. 1 copy matches pretty closely, but is truncated. And so that's a pretty obvious gross rearrangement, or in this case, deletion of chromosomal material. As chemistry, biochemistry, molecular biology, genetics has advanced, we have different tags, different labels that we can use on DNA now for different reasons. So in some cases, we would talk about a technique like this one called chromosome painting. This is where a particular chromosome would be labeled with a fluorescent tag. And we can do this now more specifically. So we can tag specific DNA sequences, specific structures. In this particular picture, the DNA itself is generally stained blue. The centromeres here are stained in red. And then we have this chromosome. I'm not sure what exact dye they're using here, but um, some chromosomes here have been stained with an additional stain. Now, if I had to guess, I would say that this is a hexaploid organism, and those are 6 copies of the same chromosome, but that's a little bit of speculation here. The point is we have this technique that is more specific and can give us more information. We have lots of different tools that we could use, different specific dyes and probes. And to that point, here's a fantastic picture that I like to look at. This is, again, chromosome painting. Now we've got lots of different stains and in some cases false color imagery just to make it look better for us. But in all of these cases, what we're doing is we're taking that chromosome spread, we are coloring it, banding it, labeling it in some way, and then lining up the chromosomes next to each other. This is a human karyotype. So we can see the 23 pairs of chromosomes. This happens to be a human male. Down here we have an X and a Y. And what you can tell on this one is that most of the chromosomes have patterns that match pretty well. There might be a little deletion here on chromosome 4, 5. It's hard to tell. I don't think so, but we can see this change that's occurred on chromosome 1. And that is, if you imagine the pattern here, yellow, red, purple, red, black, we now see that part of this is reversed. So still yellow at the end, but now black, red, purple instead of purple, red, black. This is an inversion. And that's pretty obvious here. That probably wouldn't have been as obvious with an older technique. It probably bears mentioning at this point that human chromosomes are numbered such that 1 is the longest, down to 22, which is the shortest, and then the X and Y chromosomes are usually presented at the end of the karyotype. If this was a female, we'd have 2 X's, male, X and a Y. We'll see some examples of some of the non-disjunction in the sex chromosomes, similar to what Bridges saw in the fruit flies, but we will see that here as well for humans. In this case, this technique can visualize gross chromosomal changes like translocations, where one piece of DNA is moved from one chromosome to another. For instance, if this end of chromosome 10 had some red stuff on it, and that red stuff was missing from chromosome 11, we could say that that was a translocation. We could have a piece missing, like we saw even in that crude staining a couple of slides ago. We could see a duplication, an inversion like we see here. So gross chromosomal movements are pretty easy to see with this kind of resolution. This is the fluorescent method, abbreviated as FISH, or fluorescent in situ hybridization. In situ just means inside, or in this case, in the cell. So what they're showing on this slide is a couple of things. On the left 2 pictures, we see sort of a standard karyotype kind of a view where now, in addition to the chromosomal level staining, we have something stained specifically. And so in this left picture, we have the blue overall chromosome, and then we see some specific locus stained here. Okay. Again, I'm not sure exactly what they were staining in this picture, but you can see these dots of additional fluorescent staining to show us relative position and number of those sites, whatever those are. In the middle, in the red picture, we see what looks more like a standard karyotype spread. Again, with the DNA now stained in red. And then it looks to me like something having to do with the telomeres has been stained here, because we see that fluorescent dot towards the end of each linear chromosome. Again, not sure of the specifics that went into this particular image, but that's kind of what we're looking for. This particular kind of staining, where we're staining telomeres, makes it easy to see if we've had a deletion from an end, because you may be missing a telomere. You might see that. And then here on the right side, we see more of an in situ hybridization. So these would be chromosomes actually still inside the cell, inside the nucleus. And then it looks here, this obviously isn't human. We have loads and loads and loads of little pieces of chromosome. Well, I don't think it's human. I guess we'd have to count. But again, we can see here that the DNA is stained in red. Looks like the centromeres in this case are stained in yellow. And then we see a subnuclear body here that could be visualized that's containing some of those DNA pieces. So this is a link down here. You can check out more about fish if you're interested. But in basic, here's the human karyotype. Again, this is a genotypically normal male. We would depict the karyotype like this in the title. So 46 is the total number of chromosomes, and then XY for the sex chromosomes. If something was missing, we had a deletion somewhere, we would say, for instance, 45, maybe XY or XX if it was a female. If it was an X chromosome that was missing, we might say 45X or 45X0, something like that. We'll see some of that lettering here in a few minutes, but this is how karyotypes are typically indicated. And then if there's something specific you'd like to indicate, you could put that after the standard genotype here. So deletion in some particular region or something like that. Here again, we're seeing chromosome 1 is the longest, and we decrease down. Now notice that from a gross perspective, some chromosomes have the centromeres kind of towards the center, as the name would indicate. Some have it closer to the end. That's perfectly normal. It's just that the different chromosomes have different structures. The shorter arm of a given chromosome would be called the P arm, and the longer arm is the Q arm. I think I'm getting ahead of myself in the slides, but that's perfectly normal for those to not be symmetrical necessarily. What we'd be looking for in a normal individual is that both copies of the chromosome look fundamentally the same. Again, we don't have any gross either differences in chromosome count, like a trisomy, for instance, or missing or deleted P's or inversions like we saw before. These high-resolution techniques, like FISH, have allowed mapping of what's called genetic synteny. Synteny is conserved gene order. And what we see here, this is from a study done at Oak Ridge. What we see here is the mouse sort of cartoon karyotype over here on the left. And what they've done in this figure to make it visual is they've color-coded the mouse chromosomes to reflect the locations of similar pieces of human DNA. So as you probably know by this point, humans and mice share a considerable amount of our DNA sequences. What this is showing is synteny. So for instance, we can see here chromosome 1 is shaded in this dark green color. A significant chunk of the mouse chromosome 1 is also dark green. You see this large block that's basically conserved sequence from our chromosome 1. Similarly over here, mouse chromosome 12, there's the 12, contains a large block of DNA that maps to our chromosome 14. And you can see those sort of mappings. Obviously they're shuffling, okay, fine, but what's interesting here is that there are large pieces of chromosomal DNA that are in the same order. So not just that we have the same genes, or not just that we have the same, sort of genetic patterns sometimes, but we actually have physically similar pieces of DNA. The other sort of way that we can look at these is by what's called an ideogram, and this is kind of a cartoon diagram. These would have been hand-drawn originally, way back in the days of early microscopy. Later, these could be photographed and sort of mapped out that way. Now, we'll see them as these cartoon representations to accompany digital databases of genes and things like that. So here's the human chromosome 5. It's laying on its side because it fit better on the slide that way. The centromere is marked here for you. And as I mentioned a minute ago, the short arm of the chromosome is always labeled the P arm. The longer arm is labeled the Q arm. And then those regions are numbered from the centromere to the end. And you can see the numbers across here. Think about this, though, as almost like a hierarchy, right? So we're saying in this case, this is human chromosome 5. Maybe I'm talking about the Q end. And then if we look at that Q arm here, we'll see that all of this information is listed. The first digit is a 1. And then from here over to here, the first digit is a 2. And then from here to the end, it happens to be as a 3. Think about that in terms of the evolution of our resolution, like we've been talking about. So imagine that this arm of chromosome 5 on some older technique showed up as 3 distinct bands. Maybe back then we would have said, this is the Q1 region of chromosome 5, this is the Q2, and this is the Q3. And then as we got more and more resolution, better dyes, higher resolution cameras and microscopes and things like that, then we could subdivide those. So now region Q1 could be defined into 1, 1, and 1, 2, and 1, 3, and 1, 4, and 1, 5. And then further within that, now we can go beyond, right? Not just 1, 2, but now 1, 2.1, 1, 2.2, 1, 2.3. So that's how they're read. So if you saw 5Q33.1, you would say it just like I did there, 5Q33.1. And of course now with genome sequencing and stuff, we can just say what base pair we're starting with, but you'll still see this kind of banding pattern being discussed. Speaking of digital databases, the National Center for Biotechnology Information, or NCBI, as part of the National Institutes of Health, does have a map viewer where you can go and look at all sorts of different species, and this link points to the human genome version of that. But when you go in there, you'll see that ideogram like we just talked about here on the left, and you can zoom in to what level you want to talk about. You can pick across the top here, chromosome 1 through 22, or X or Y. MT would be mitochondrial DNA. And then you can keep zooming in. So you'll still see that labeling like we've talked about. So, the different parts of the chromosome over here, you can zoom in and then see which genes are there. When you go beyond that, these links are all to different sub-databases under this, so you can get more information. Over at the far right, you would see not only where that gene is located, but if we know what it does, you would see, its name over here. You can click on any one of these genes and it will open up a whole page about probably way more than you'd like to know about it, but what we know about it, its sequence, if there are any variants, expression levels, all kinds of stuff. It's really a fantastic database for genetics in the modern world. So back to some of our terminology. As we're looking at those cytological presentations, if an organism looks normal, whatever normal means for that organism, we would say it's euploid. Remember, eu is this prefix that means kind of true or accurate or correct. So this is a euploid organism. So for humans, we should be diploid. If we've got all of our little chromosomes in there like we should, we'd say that's a euploid organism. An organism could be triploid, maybe. Triploid organisms are often sterile, but several of them have been found in nature to survive just fine. That would be a 3N kind of a scenario, 3 copies of each chromosome. If that's what they're supposed to be, then fine. A euploid organism would have, triploid organism would have 3 copies of each chromosome. You could have a tetraploid organism as well, or hexaploid or octaploid, whatever it might be. Different organisms have a different arrangement, but whatever it's supposed to be, we would use the term euploid. And I just want to point out here, anything greater than 2, we can lump generically into a polyploid kind of a word. Okay, just meaning more than 2. Why 2? Well, because humans are diploid. So We're a little human-centric here. Aneuploid, or not euploid, is anything other than euploid. So if a piece of a chromosome is missing, or a whole chromosome is missing or duplicated, or something is flip-flopped around in a translocation or an inversion or something, all of those would be aneuploid states. Something's wrong, something's different to normal. And we talked about some of those rearrangements that can sometimes occur. Talking about polyploidy, this is super common in plants. So about half of all sequenced plants or studied plants are polyploid. About 2/3 of those that could be considered grasses, so things like, obviously grasses, but also things like wheat and rice and things like that. About 2/3 of those are polyploid. These tend to be more robust. Remember that the more copies of a gene you have, typically the more resistant to random mutation that gene would be. And so a mutation showing up wouldn't necessarily impact the function of the plant unless it showed up multiple times. Also, polyploid cells tend to be larger than diploid cells. In fact, the higher the ploidy, typically, the higher the size of the cell. They also tend to reproduce asexually. That's not 100%. But they often do, especially if we're talking about having a non-even number, which is possible, not common. So shown here are chrysanthemum, which is a tetraploid organism. Cotton plant is a tetraploid as well. Banana over here is a triploid. And then maybe you've done a DNA extraction experiment at some point, some demonstration or something like that. We do that from strawberries quite often because they're octoploid. They have a lot of DNA in their cells. Getting to that idea of them often being sterile and having to reproduce asexually, we can see a picture here. And this is showing us now a triploid organism. So whichever chromosome it is that we're looking at here, we have 3 copies of it. Those have already been duplicated in these pictures, and so we're right into mitosis. The chromosomes are lined up on the metaphase plate. And what they're showing us here are 2 different arrangements that could happen at that point. We could imagine them perhaps, I mean, they might arrange normally, okay? But they could arrange like this, in which case during anaphase, we might get 2 copies pulled in one direction and 1 copy pulled in the other direction. That would give us, instead of a functional triploid later on, it would give us something else, 2 different kinds of cells. Or if we had them all pairing up by their centromeres, which, common arrangement at mitosis, those could all be pulled in the same direction instead, giving us a null cell and an overstuffed cell at the top there. So the point is, when things don't line up evenly and symmetrically, meiotic pairing is uncertain. And so we can get this sort of non-disjunction event happening pretty easily. There are some examples of fertile polyploids in the book, and actually I'm not sure if it's still page 116. I guess it depends on which edition of the text you're looking at. But there is this, what they call a plausible mechanism for the origin of a fertile tetraploid. Now, stick with me here for a second. What this is saying is that over sort of evolutionary time, top to bottom, we had some diploid plant and some closely related diploid plant that were different. So here the letter is not meant to indicate a specific gene or a specific allele. This is meant to indicate an entire genome. Okay, so diploid plant over here has 2 copies of the A genome set, whatever that looks like. And this similar but different diploid plant has a different genome set. And if those happen to fertilize one another, we would get a hybrid, an AB hybrid. And this thing would have a very hard time during meiosis trying to line up those chromosomes correctly, right? Because the chromosomes from the A genome wouldn't necessarily match very well to the chromosomes with the B genome. There could be different chromosomal structures and things like that. And so this would be, as a result of that cross-species mating, this would be probably a sterile hybrid. Well, now what if we had a complete non-disjunction event? So this is a chromosome doubling. This is super rare. Okay, I don't want you to think this happens all the time. But if this happened, if for some reason a cell went through the beginnings of, say, mitosis, doubled all the DNA, but then didn't divide, we would end up with a cell that now has 2 copies of the A genome in its entirety and 2 copies of the B genome in its entirety. So the book labels this as a fertile tetraploid, and depends on how much you want to split hairs here about whether that's really a tetraploid, since the A genome and the B genome aren't exactly the same. Um, maybe you'd call it a diploid of diploids. I don't know, but the point is we're back to an even number of genome sets. And so now in this case, this is what we call a rescue step, and then this thing could go through normal meiosis or mitosis and so forth. The gametes from this organism then would have 1 copy of the A genome and 1 copy of the B genome, such that when those 2 gametes came back together, we'd have 2 copies each, AABB. This is thought to have happened with wheat. Modern wheat is a hexaploid, and it's thought that this happened sort of a couple of times over, the evolutionary time scale of that plant. Couple more words for you, add to your vocabulary list. If that polyploidy happens between 2 different species, like we just saw here on the previous slide, we would call that allopolyploidy. Right, allo meaning not the same. So those are created between different species. Autopolyploids created by chromosome duplication within a species. And here's that wheat example. So again, using these letters to represent entire genomes. All right, so it's thought that some historic wheat precursor strain was a diploid here with 14 total, but 2 copies of each of 7 chromosomes. And some other strain, B, had the same deal. When it did that cross-species mating, it's thought to have given rise to now a AB haploid, hybrid organism with a haploid number of 14. Now, I like this labeling a little bit better because this isn't a true diploid, right? Because I don't have 2 copies of the A genome or 2 copies of the B genome. So it's not really a true diploid. So what we're saying is it's got all these chromosomes now and a haploid number of 14. So 7 A's and 7 B's, if you're going to think about it that way. It then can go through that rescue step. It's thought to have done this rare chromosomal genome level duplication. Notice the haploid number stays the same. There's still 14 total pieces of DNA required to represent this whole thing, but now we've got more copies of that. This would have been a fertile, I guess we could say tetraploid hybrid that could go on and do its thing for a long time. Okay, this diagram sort of loses that time scale. Eventually, though, if we imagined a cross-species mating happening again between this species and a related one, we could imagine now the same kind of thing happening a couple times. And so our modern bred wheat, this is probably cut off on the video, it says down here n equals 21. Modern bred wheat is a hexaploid with a haploid number of 21. So there are 21 pieces of DNA in that genome that represent an entire genome, and there are 6 copies of that thing in the, or 6 copies of each gene, I guess you could say, in that modern species. So a hexaploid hybrid. So this is sort of the idea of how it's thought to have happened. Again, we're talking about a lot of time, and we're talking about really rare events. So evolutionary timescale. In humans, tissue-specific polyploidy has been identified. So polyteny here, we're going to see in a minute with some fruit fly stuff, but polyteny is where a part of a chromosome duplicates again and again and again. So rather than duplicating an entire genome, we duplicate a part of a chromosome. So this is a process called endomitosis. So the chromosomal replication happens, separation of sister chromatids happens, but then we don't actually divide the cell. This has been shown to happen in some human liver and kidney cells that are actually found to be tetraploid. And so you can read this quote here. I won't read all of this, but I just want to highlight that in adults, 70% of all hepatocytes, all liver cells, um, are shown to have been tetraploid in rodents, but still about 40% in humans. And this was referenced out of a couple of papers down here at the bottom. But it's not obviously our normal. Humans are normally diploid with haploid gametes, sperm and egg cells. But there are instances where we've seen this, what's called endomitosis. And here's that polytene stuff that we just referenced on the previous slide. So in Drosophila, and this I hope you can tell is an older sort of technique, but in Drosophila, polytene chromosomes are produced here by multiple rounds of replication without separation of the sister chromatids. And so what happens is that you get this chromocenter. This is where all the centromeres sort of coalesce, and then the multiple copies of the chromosome all sort of span out from there. Kind of a cool structure. These things are big enough that they were identified a long time ago. And see here, this is C.P. Bridges again. So this is getting back into Thomas Hunt Morgan's lab and then later Bridges' work. But these could be seen very easily, even back in the day. And so you could look at chromosome spread and see, based on the banding patterns, which are sort of generally represented here, you could say, oh, I see that this region's been duplicated or triplicated or multiplicated, lots and lots of copies. These large chromosomes allow us to really get in there and look at gene positioning and look at sequencing, and we can see all sorts of stuff. So again, the language here, aneuploidy is anything different from normal. Within that, we might think about having specific numbers of chromosomes. So having an extra chromosome in a diploid would be a trisomy. Having only one copy in a diploid would be monosomy. We could talk about hypo and hyper as prefixes to talk about any piece of a chromosome being changed. Studies have been done in lots of different organisms. And this is one, Datura, that you will see. And I want to just sort of preface this a little bit. When you read in the textbook, they'll talk about this thing. This is the seed pod from this plant. It's sort of a spiky little green thing. It's normally a diploid plant. And so up at the top, they show you what a normal seed pod should look like. And then each of these pictures is showing you a trisomy of that one chromosome. So if I had a diploid organism that had an extra copy of chromosome 1, so it was trisomic for chromosome 1, its seed pod apparently is smaller, but generally the correct shape. If I have a trisomy 2 instead, it's smaller and has a different shape. Trisomy 5 and 8 and maybe 10 are almost normal looking seed pods. So apparently the implication is that whatever genes are involved in seed pod formation are not located on chromosomes 5 and 10, and I guess 8's a little different. But you can see the variability here based on what is being triplicated. Now, obviously, having an entire extra copy of a chromosome, that's a lot of genes. In this organism, it's a 1/12 of the genome then that you're overrepresenting. That's a lot. So, the point of this slide, I think, is just to show us that the effects are variable, but oftentimes can be sort of withstood by the organism. Now, in humans, there are only a few known trisomies of our autosomal chromosomes. And of course, one of the most common ones is trisomy of 21, which leads to a wide-ranging pleiotropic condition called Down syndrome. We see a Down syndrome karyotype on the right. So we see normal pairing of chromosomes 1 through 20, and then we see that extra copy of 21. If we'd done this by fluorescence or some other technique, we could confirm that these were 21, but someone skilled in looking at these karyotypes would arrange them pretty easily. We can see this particular individual is genotypically female. We see the 2 X chromosomes there. So in humans, there are only a couple of trisomies that have been shown to exist. And typically, those occur by non-disjunction. Well, they always occur by non-disjunction. Just like we talked about for fruit flies, we typically think about the egg as being the non-disjunction gamete. It's not because it's special so much, but more because it is more of the rate limiting step. If I drop into chemistry for a second, lots and lots and lots of sperm are produced for every egg that's produced. And so an error in a sperm is statistically likely to be unnoticed, whereas an error in an egg, statistically more likely to show up. The point of this picture is to remind us of the two steps involved in meiosis, meiosis 1 and meiosis 2. If we have a non-disjunction in meiosis 1, all of our gametes will be affected. And you can see why that would happen. So if the lavender and dark purple here, lavender and grape, I don't know, if they stayed together in meiosis 1, then one of our intermediate steps would have all of our chromosomal material and the other one would have nothing. And so if then meiosis 2 is normal, we get 2 diplo 21s and 2 nullo 21s, if we're talking about chromosome 21 here. From this picture, you can start to tell maybe that's not really 21. It doesn't have so much of a P arm on 21. But then if one of these diplo eggs was fertilized by a normal sperm, of course, then we would have 3 copies of 21, 2 from the mom, 1 from the dad. So non-disjunction in meiosis 1 results in no gametes being normal or euploid. Whereas if we had non-disjunction happening in meiosis 2, that first division goes off without a hitch. Then the second one is normal in some cases, non-disjunction in some cases. Now we could end up with normal chromosomes, normal gametes in about half the cases, and aneuploid gametes in the other half. And that's 1/4 diploid, diplo 21 here, and 1/4 nullo 21. And so if this were the case for somebody, they would have a decent shot at having a normal child here. You'd say sort of a 1 in 4 chance of having a Down syndrome child, and the nullo 21 would not likely be viable. And so I mentioned a couple of times now that there are only a few human trisomies that have been observed. And this table is showing us a bunch of them that have been observed, but from our autosomes, just these 3 at the top. So we mentioned Downs. It would be represented as 47, and then we would say either XX or XY, and then we would indicate a plus 21. So the extra one of our 47 is a 21. That clinical syndrome is called Downs syndrome. Estimated frequency according to this table is 1 in 700 births. Phenotype over here shows that pleiotropy I was talking about. Lots of different aspects to it and a wide range of severity. If we had a similar trisomy but on chromosome 13, that's been called Patau syndrome. It's more rare than Down syndrome, but we can see, again, a pleiotropic collection of phenotypes. And the last one is chromosome 18 trisomy. That's been called Edwards syndrome. Again, more rare than Downs. But here you can see this one's pretty severe. 90% die within the first 6 months after birth. Something about having that extra 18 is pretty bad. Now, you'll notice that if you look back at our karyotype pictures from before, remember that chromosome 1, 2, 3, those are the biggest chromosomes. And by the time we get up to 18, 19, 20, 21, they're pretty small. So these are the only 3 that have been observed clinically in humans. There have been no living trisomy 1s or trisomy 2s, for instance, that have been observed. And the theory here is that it's just overrepresenting a too large piece of the genome. Just can't be tolerated. And those are embryonic lethal. We just never see them develop. Not to say they don't happen, probably do, but we don't see them develop into something we can observe. However, when we talk about sex chromosomes, it seems like the sky's the limit. You can see at the bottom down here, there are all sorts of bizarre non-disjunction events that can lead to all kinds of weird chromosomal arrangements in the sex chromosomes. The one at the bottom, the Ch.06 syndrome, is having that triploid trisomy in the X chromosome, so 47XXX. That's got about the same frequency as Downs. And notice over here, a female with usually normal genitalia, but limited fertility, right? So remember what we talked about for sex differentiation in humans, that if you have that X chromosome without a Y chromosome, the developmental path for the embryonic gonad would be towards female um, uh, genitalia, female tissues, all of that. So that explains this first part. The second part, limited fertility, we have now 3 X chromosomes. And so you can imagine what that would look like when you're trying to go through meiotic divisions. Some of your eggs would be aneuploid. And so that would lead to that limited fertility. Up here, the other common ones you should be aware of are 45XO. So missing a copy of, well, the second sex chromosome, be that X or Y. Typically, this forms from having one of the gametes deliver the X chromosome and the other one having been a non-disjunction chromosome that didn't get the Y, or the other X, I guess. This is called Turner syndrome. They mention here 1 in 2,500 female births, right? We wouldn't ever have a male with Turner because there's no Y chromosome. So this is a female with limited sexual development, generally sterile, and then pleiotropic effects otherwise. Similarly, if we had an extra X chromosome, that's starting to look a little bit like what Bridges did in the fruit flies. If we had 47XXY, this is called Klinefelter syndrome. These individuals are male now because they have that Y chromosome. They're male, but again, subfertile. All right, they've got a somewhat overexpression of the X-related genes. So we tend to have a decrease in male secondary characteristics and an increased level of female secondary sex characteristics. So again, these are unusual in the sense that, you can see the statistics there, but the point I think to get across here is that although we saw for this plant that a trisomy in any of its chromosomes is tolerated, that's not true in humans. We have a limited number of chromosomal abnormalities that we can tolerate. And often with pleiotropic effects, because remember, we're increasing the expression of an entire chromosome's worth of genes. And so that has a lot of impact. Here's that picture of Turner syndrome. So if we had a null X egg, for instance, non-disjunction gave us a null X here. A normal sperm fertilizes that. That's an X sperm. And we get a 45XO Turner syndrome. And you get the entire individual having that genotype. The other way this can happen is a normal egg and a normal sperm coming together to form what would be a normal female zygote, 46XX. And then we end up with a loss of an X chromosome in that first or one of the early somatic divisions. In this case, we would have a chimera, where the tissues that develop from this part of that organism would be normal, and the tissues that develop from this part would be Turner, would be aneuploid. So a somatic mosaic, this is called. And again, as we've gotten better technology, we can map certain conditions to certain regions. Like we saw, obviously, the trisomy 21 is Down syndrome. That's pretty obvious on a karyotype. This is one called Criticot, a syndrome where the person, this is French, and I probably butchered it, for a cry of a cat. The sound made by this human individual when they're an infant sounds like a cat's sort of screeching or meowing. This could be represented as 46, in this case, XX, female, and then 5p negative, or so showing us we have a deletion on the 5p arm. Or we could say specifically delta 5p14. Depends on when you were talking about it as to how good your resolution was. Using that telomere mapping that we talked about before, that kind of fish technique, we can see that this copy of that chromosome is missing the end. certainly missing the telomeres. It looks like it's missing a bit more. So using cytological techniques, we can map certain conditions to certain regions of chromosomes. Bridges has been doing that. People have been doing this in fruit flies since forever, since we started looking at DNA structure. These are easily observed, and you can see here's a normal polytene chromosome at the top. This is meant to show you the region here. If we deleted some of it or had what's called an inverted duplication, we can see sort of this bulge of extra stuff. Again, lots of work has been done in fruit flies, and we saw some of this with the fruit fly eyes and legs and wings and all of that in the textbook. What we can see here is that there's a particular mutation called the bar mutation. It makes the eye look, instead of sort of roundish, makes it look like a bar, like a vertical slot. In a normal fruit fly, it would sort of, the chromosome would look like this. If that region is duplicated, and this is the 16A region that we're talking about right here, is duplicated, we get this bar. And if it's triplicated, we get what's called a double bar, so it's more severe. So we can impute from this that whatever controls the shape of the eye is represented by a gene that's in that segment, and that the higher expression level of that thing, whatever that thing is, causes more and more of this genotype to occur. Now, of course, we could go in and genetically sequence it, find the gene, and try to figure out what's going on. And that work has all been done as well. So I've used some words along the way. Inversion is reversal of a segment. It can be peri or paracentric, depending on whether the centromere is involved or not. Translocation is movement of a piece of DNA from one chromosome to another. This can occur in a non-reciprocal way, which means we've lost something. So I moved a piece from one place to another, but lost a little bit along the way. Or reciprocal, where I don't have a loss of material overall. And in some cases, we can see compound chromosomes where entire chromosomes get fused together. And this is usually pretty bad. So we end up with two centromeres. And again, those normal processes of mitosis and meiosis are challenging. Here's a diagram of those two types of inversions. So a pericentric includes the centromere. And so this, just for ease of use here, is just lettered all the way across rather than using proper lettering and nomenclature. But what they're showing us is that this region between E, F, and G got flipped around. And so showing it in lighter purple just to show you what got flipped around. Because it includes the centromere, that's a pericentric inversion. A paracentric inversion, and I'm sorry I didn't name these things, but this excludes the centromere. So some chunk here of the, in this case, the Q arm got flipped around. Okay. Does that matter? Well, not usually, or not often. It depends on what's going on. So if you can imagine here that this, whatever gene E was, maybe was driven by a promoter region. We'll talk all about that stuff in the second half of the class, but you probably remember from Bio 101 type stuff that genes have control regions that make them turn on and turn off at certain levels. If the control region for gene E was located right here, and now that control region is driving something else, that could be a problem. It would affect what's going on with this gene G. It would affect the level of gene E. That could be real bad. Here's one way that this can be allowed to occur in the normal process of mitosis, meiosis, where the chromosomes need to line up against one another. If one of the chromosomes was inverted, they can still form a homologous pair, and that would be shown here. So we would have our one chromosome, the inverted one, shown on the top now. The bottom one can form a loop like this and still get the proper pairing. Notice that bottom one, A, B, C, D, E, F, G, H in order, but forming a loop. So they can still pair, they can still propagate. Meiosis and mitosis can carry on. And in fact, inversions are not altogether uncommon in the general population. It's just a matter of what got inverted and exactly where as to what the effect might be. Translocations, we mentioned the definitions here, reciprocal and non-reciprocal. Here's the way that those chromosomes could pair. So here would be an example of a reciprocal translocation. So this end bit of the purple chromosome got switched with the end bit of the green chromosome, but notice we haven't lost any genetic material overall. So again, this might be detrimental, depending on where the control regions are, exactly where that break occurred, but it's not unheard of to have translocations that don't have a phenotypic effect. And then, of course, if we were trying to separate these now, we don't have perfect homologous pairing anymore. But if we looked at pairing them up in this way, just lining them up on the metaphase plate like this, now we can have that proper pairing. And then in that case, it depends on how exactly these things get separated. So if we start talking about disjunction here, or that separation, right? Imagine we have this line up here. We could have a disjunction like this, and the letters A, B, C, D are just meant to represent regions, okay? But if we had a resolution of that in this vertical format, we would have the cell on the left getting 2 copies of D and no copies of B, and vice versa on the right. All of these gametes would be aneuploid. If we have adjacent disjunction in the other direction, we'd have the same problem. Now here, 2 copies of A and no C going up, or 2 copies of C and no A going down. All of these would be aneuploid gametes. What if we had this alternate disjunction, it's called, sort of a diagonal alignment? Now, although the pieces are mixed up, we'd still have an A, a B, a C, and a D in each cell. So these gametes would be normal, although the chromosomes constituting them would be reshuffled. They would show that translocation. Okay, so again, there's a little bit of randomness here depending on how things line up at the metaphase plate. But once we get through this, then the gametes from this would be just fine. They've got all the genetic material they need. And then compound chromosomes. This is that sort of bad time that I mentioned. Typically, we end up with a problem here because the 2 chromosomes now are fused at their centromeres. This is in Drosophila, again, a condition called attached X chromosomes. The Drosophila X chromosome it does have a P arm, but it's very, very small. The centromere is near the end. And so what we're seeing here is a female that's gone through this compound chromosome formation. And if she had a mutation, this little mutation M we can follow, we can see because these 2 chromosomes are now the same chromosome, technically, they're a compound chromosome, 1 centromere means that they go together wherever they go. We'd get an egg that was double X and an egg that was null. And then during fertilization, we would get this triple X in 1 box of our Punnett square. That's typically lethal. We get the YO with no X chromosome. That is also lethal. That's what Bridges saw before too. We could get an XXY, the exceptional female. Remember in Drosophila, it's not about the Y chromosome. It's the balance of X's to autosomes. So having 2 X's is a female. Or the XO, sort of the Turner's equivalent, although they don't call it that in flies, which would be that exceptional wild type male. So you can work through the genetics on this. This would be for a mutant female and a wild type male. And we can see how this works out. But the key thing here is that these 2 chromosomes are now technically actually 1 chromosome, right? So they never would separate again. If we have that in humans with some of our what are called acrocentric chromosomes, so these are the chromosomes that have the centromere towards an end. This is what's called a Robertsonian translocation. If this occurred, that we had this sort of crossing over event, this fusion happening at the centromere, remember all centromeres have a similar kind of a structure, so this isn't unheard of. If this happened, you can see we'd get 1 quite large chromosome and 1 really tiny chromosome. And generally speaking, that tiny little fragment would be lost over time. So we'd have a loss of this genetic material, like the P arms of these 2 acrocentric chromosomes. And so as we go through this slideshow and we get here to the end now, I hope what we've been able to sort of look at is the cytological techniques that were used throughout history and how they've evolved. I didn't talk specifically about DNA sequencing, but that's where we are now. So we can still do cytological stuff and then sequence what we need to. And then we've talked about all of these exceptions to the euploid state. So polyploidy could be completely normal in an organism. We talked about triploid and tetraploid and hexaploid organisms. That's fine. But if an organism is supposed to have one ploidy and for whatever reason it has another one, that's a problem. So under aneuploidy, we had all the things with translocations and deletions and additions, duplications. So lots and lots of different versions of that. And then at the end, talking about rearrangement of chromosome structure. So consider this sort of a bridge now between the original sort of work that emphasized the chromosome as the inheritable material, and what we'll see in the next chapter where we start talking about specifically how the DNA moves and how movement of one piece of DNA might affect movement of another. So for now, check out the textbook, go through the slides, and practice up with some problems in the book. And I will see you again next time.